if you can't hear me now, can you please type in the chat box? Okay, so it seems like everyone can hear me. Welcome to our second seminar of test for immune activation by your therapeutic oligonucleotide, as given by our OTS president, Dr. Krieg. If you have questions during the talk, please type them in the chat box, because unfortunately it's quite difficult to handle 60 to 100 attendees with questions, and I have to permit people to speak. So that's not very effective. So if you have questions, please put them in the chat box. Okay, Art, over to you.
Yes, thanks very much, Petra. I appreciate that, and thank you all for dialing in today. I will be talking to you about, no, I lost it. Okay, good. I'll be talking about testing for immune activation. And first of all, on disclosures, I do want to point out I did discover the stimulatory effects of CpG DNA many years ago. And so from a scientific perspective, I'm really excited about that. And I'll try to give a completely neutral presentation today. But I also want to disclose I have stock in Checkmate Pharmaceuticals, which is developing those for therapeutic applications. But today our focus is really on screening for the immune effects of nucleic acids. And this is a brief agenda. I'll first give a very general introduction to why the immune system detects nucleic acids at all, then talk about the immune pathways that may be activated by them, how to screen for them in vitro, and how to screen for them in vivo, and what can happen in vivo, including in human beings. Is going to be on how to detect immune activation. Now, identifying the mechanism of action for immune effects that you see with an oligo and avoiding those effects is really a different topic that's out of scope for today, but that potentially could be a focus for a future webinar. Now, this is a slide, a picture taken from an excellent review article by Gunter Hartmann in Advances in Immunology last year. And it makes the point that of this concept of nucleic acid immunity, that is that every organism on Earth, in order to maintain genomic integrity, has to be able to avoid external nucleic acids coming in and basically replacing the endogenous nucleic acids. Viruses, retroviruses, inject their genetic material into other cells. And if you don't have any defenses against that foreign genetic material, you're an evolutionary dead end. And so as far back as bacteria and very primitive organisms, there are mechanisms to prevent foreign gene expression. Nucleases are one type of these. The CRISPR-Cas system, of course, another great example. And as you go up the evolutionary chain to eukaryotes and to mammals, there's increasingly sophisticated defenses to detect foreign nucleic acids and drive defenses against whatever the pathogen may be that contains nucleic acids. Now, from a therapeutic perspective, the consequence of this is that we are trying to sneak our therapeutic nucleic acids into cells without activating these defensive systems. And that can be very challenging. And so what I want to talk about today is just how some of these different defenses work and how to detect when they may be activated. So what immune pathways may be activated? DNA sensing immune pathways are shown in this rough schematic from Martin Schley and again Gunter Hartmann, making the point that there are several different pathways. This is the cell that we see, and the nucleus of the cell is at the bottom. There are toll-like receptors in the endolysosome. In the cytoplasm, there are several different factors, but most notably STING, the C-gas STING pathway, where C-gas detects foreign double-stranded nucleic acids or endogenous double-stranded nucleic acids. So in a setting of stress, where you have cells where the nuclear integrity is lost, and DNA leaks out from the nucleus into the cytoplasm, that also can activate the C-gas system. So this is not just a pathway for detecting foreign nucleic acids. It's also a pathway for detecting cellular stress or perhaps tissue damage. Now, there's an important species-specific difference in the C-gas sting pathway between humans and mice. In mice, the pathway really predominantly activates this IRF3 interferon-related factor pathway that you see down below the sting. But in humans, I drew a red arrow over here to the inflammasomes because there was a recent paper in Cell, the reference is at the bottom, showing that in human cells, but not mouse cells, the inflammasome pathway is also activated. So this is a very important species difference. This is a theme I'm going to be coming back to, and it really limits our ability to extrapolate from mouse systems into humans. And it's something when we're screening for the effects of nucleic acids, we have to keep this in mind because the results of screens in mouse cells may give different outcomes than what we get when we're screening with human cells. Now, the other major DNA sensing pathway that I'd like to talk about today is the TLR9 pathway. TLR9 detects the so-called CpG DNA, unmethylated CpG dinucleotides that are present in bacterial and vertebrate DNA, bacterial and viral DNA, but are not present or are methylated in vertebrate DNA. I shouldn't say they're not present, they're suppressed. Now again, there's an important species-specific difference here. Mice express TLR9 in many different immune cells, monocytes, several types of dendritic cells. But in humans, TLR9 is more or less only expressed in plasmacytoid dendritic cells and in B cells. And so the consequence of that is that we get different outcomes in terms of the cytokines that are produced between mice and humans. And I want to point out also that whereas with the sting pathway, there is interferon production, it's relatively low compared to the very high interferon pathway through the TLR9, high interferon induction through the TLR9 pathway. And conversely, IL-1 beta is highly induced by sting agonists in human cells because of the inflammasome pathway, but relatively little IL-1 beta is induced in human cells. And I see there is a question, is the pathway human or primate specific? And the answer to this pathway that shown on this slide here is, as originally published, more applicable to rodents than to humans, with this correction that in humans the red arrow does activate inflammasomes. And in general, primates and humans seem to be very similar, but all of this is something that's evolving has been evolving over the last years. This Cell paper just came out a few months ago. Now, if you want to detect immune stimulation occurring with DNA oligonucleotides through these pathways, you can detect it pretty well in vitro using interferon alpha and beta as a screen, just test for induction of interferon alpha or beta, or the interferon inducible factors, IP10 or CXCL10, and a variety of other factors. That will show you activation that's occurring the IRF pathways, the interferon response factor pathways. IL-6 and TNF-alpha are induced by NF-kappa B, which you can see down at the bottom of the, whoops, I crossed it out, oh well. NF-kappa B leads to induction of IL-6 TNF and a variety of other inflammatory cytokines. And then IL-1 beta, testing for that, will reveal the activation of the inflammasome pathway. There are certainly other factors that you can test for as well, but it's not necessary to test for tons and tons of factors. You can have a fairly focused screen and be reasonably confident that you're going to pick these up. Moving on to RNA, moving on to RNA sensing pathways, the principle here is the same, that there are RNA sensing mechanisms in the cytoplasm, and I'm circling some of these here, and then again in the endosomes. there are RNA sensing TLRs. For RNA, there's three, TLR3, TLR7, and TLR8. But again, there's an important species-specific difference. TLR8 in mice doesn't really seem to be functional, at least in terms of signaling. It does seem to have some kind of biologic effect, but it's not that well understood. Now, turning to the cytoplasmic RNA receptors, the main one that I'm going to talk about today is RIG-I. RIG-I detects RNA molecules with a blunt end at the 5' and 3' ends with a diphosphate or triphosphate on the 5' end. And these compounds are often produced by in vitro transcription of RNA. And when people are making mRNA or other in vitro RNA transcripts, you can often get sort of byproducts of the reaction or anomalously folded RNA transcripts that can activate RIG-I. And this paper from Schmidt et al. Is one of the papers that points to that. This is the reference at the bottom of the slide, that points to that as a potential complicating factor of experiments where people didn't realize that these kinds of transcripts were being produced. So it's something to be aware of when you're working with RNA. And okay, good. So some quick key concepts here. Rodent data do not predict human responses. There are different receptors in the endosomes and the cytoplasm. And one point I'm not going to talk a whole lot about, but I think it's worth keeping in mind. There's some evidence, although I haven't found it published, but people have told me that they believe that some transfection reagents will preferentially deliver an RNA or DNA payload into the nucleus or into the cytoplasm. Just something to be aware of. You might want to screen several different transfection reagents when you're doing experiments where that could matter. Each receptor can detect different elements of nucleic acids. RIG-I detects the 5' triphosphate or diphosphate, and a lot of the downstream pathways are the same. Now today I'm going to focus on two of the DNA activated pathways, the TLR9 and the STING pathway, and two of the RNA activated pathways, TLR7/8. And I put those together because typically most RNA molecules will activate both TLR7 and TLR8. There's slightly different sequence specificities for TLR7 and TLR8 in humans, but in general, most RNAs are likely to activate both, and then RIG-I. Now, one other difference I'd like to mention among these pathways is that RIG-I and STING are expressed more or less ubiquitously. Pretty much all cell types will express them, and they can be activated by nucleic acids in those cells. Whereas the TLRs are expressed in restricted immune cell populations. And I make that point on this slide here, showing the cell specificity of some of the nucleic acid receptors. Here is a tumor cell on the left-hand side here, and in tumor cells, you will have RIG-I and STING expression, but typically you don't have TLR expression in those cells. There are some publications in the literature saying that there is functional TLR expression in tumor cells, and in B-cell malignancies, that definitely is true with TLR9. Not so clear in other types. Now, the effects of STING and RIG-I in tumor cells, they can induce production of some of the inflammatory cytokines, and it can also induce apoptosis, generally low production of interferons. Monocytes and macrophages also express STING and RIG-I and can be activated through those, and again, that can cause inflammation. Sub-low interferon production. And one of the biologic roles of monocytes and macrophages is tissue homeostasis. I'm afraid the formatting of this got messed up a little bit in the Mac to PC transition. Next, we have conventional dendritic cells on the, I'll just circle that here. And conventional dendritic cells are responsible for antigen presentation, T-cell priming, and they express a variety of different TLRs any of which can activate those functions in the cells. Now, a very special type of immune cell is this plasmacytoid dendritic cell on the far right. This is the one cell type in the body that is really specialized to produce large amounts of interferons in response to infection. Plasmacytoid dendritic cells really only have two immune receptors that will trigger that strong interferon production. Those are TLR7 and TLR9. And they seem to have a role in regulating tolerance and immunity. So the concept here is simply different types of cells express different TLRs. And one of the reasons this is important is sometimes I've seen in the literature that people assay for immune response to their nucleic acids by using, for example, fibroblasts. Well, if the cells that you're assaying don't express any TLRs, and fibroblasts don't have very many, then you're not going to detect the potential immune stimulatory effects of your nucleic acids. So you have to be aware of the cell types that express these receptors if you're going to detect immune stimulation occurring for them. So now let me turn to in vitro screening. What are the simplest assays that we can use to detect that? It's really not that difficult. What we do, well, what you want to screen for, going back to the points I made before, for in vitro screens, if you screen for interferon alpha or beta, you're going to pick up the IRF activation. IL-6 and TNF will pick up NF-kappa B activation. And the IL-1 beta will detect inflammasome activation. So if you have a screen that includes those factors, you're really picking up the major pathways. Now, there's nothing wrong with including additional factors in the screen. One of my favorites is CXCL10. That's a really sensitive marker for interferon effect. And in fact, in vivo, CXCL10 is much more sensitive to detect TLR activation than interferon alpha itself is. The interferons don't seem to be in the circulation nearly as much as CXCL10 and some of the other interferon-inducible chemokines. IL-12, very popular factor to secrete for, very powerful biological factor. Commonly people will use a multiplex panel. There are a number of them out there. Luminex has a 25-plex chemokine panel that covers a whole lot of different factors. But you don't really have to include all of those different factors. And frankly, I think it can sometimes be confusing to have too many different factors in your panel. The assay method, any multiplex assay will work, MDS, Luminex, any of those are fine. And the cells to screen in, we use just human PBMCs. We don't routinely use mouse cells for in vitro screening because of the species-specific differences. The human PBMCs, one point I'll make, just a technical point, is that we use much higher cell concentrations than you usually would use for in vitro assays. We're using 3 to 5 million cells per mil. Oftentimes people will just use 1/10 of that. And we have found that U-bottom plates or round bottom plates seem to be more sensitive for detecting the induction than flat bottom plates. The cells like being clumped together to respond to the nucleic acids. Of course, you should always, when you're developing a new assay, you should work out the systems yourself and make sure that you've optimized the culture conditions. And for different kinds of nucleic acids, different formulations, other culture conditions may be better. Now, an alternative to PBMCs that's widely used, especially with people developing LNP formulations of RNAi, for example, is the whole blood assay. And there have been some reports that whole blood assays may be more sensitive than PBMC assays for detecting immune effects. In our hands, we haven't found that, at least testing with antisense DNAs and in some RNAis. But you could consider trying both of those in your own case because you might have different results there. Mouse spleen cells, I consider optional. Usually, the human cells will pick up the active sequences, but certainly there's no harm in doing mouse spleen cells too, and the culture conditions there would be very similar. In terms of the timing of the cytokine induction in vitro, typically secretion starts within a few hours. We usually let the cultures go for 48 hours to just get the most sensitive detection, and we find that it seems to be a little more reproducible with 24 to 48 hours of culture. But your mileage may vary. And again, I put a reference at the bottom here for screening for different RNA motifs. In this paper, looking at the effects of TLR7 and TLR8 motifs in RNA, and making the point of how these screens can help you to differentiate different immune pathways. So now let me turn to in vivo screening. And the screens that we use in vivo, we use a rapid cytokine induction. That's just typically 3 hours for the peak cytokine induction in rodents, although I'm told with some formulations and some nucleic acids, the peak time may be 24 hours to see induction. Here again, you probably ought to test several different time points for whatever type of nucleic acid and formulation working with to make sure that you have the optimal condition. And in terms of what to screen there, I guess I left that out. I forgot to update that on the slide. What I would screen for the cytokine induction would be the factors that I'd mentioned on the previous slide, but especially, as I mentioned, I would test CXCL10 or other interferon-inducible chemokines as a marker for the interferon induction. Because in vivo in mice, you don't tend to see a lot of interferon in the circulation. And that's the same thing in humans. In humans, TLR induction is much more sensitively detected by serum CXCL10 than it is by serum interferon. Hepatotoxicity is another screen that I think anyone developing therapeutic nucleic acids wants to perform. And I put a footnote here to make the point that there are multiple mechanisms of hepatotoxicity from nucleic acids. Some of these are immune-mediated, some of them are not. I have a couple of references at the end that talk about that too. And I'm not sure that it matters. Typically, if people have a hepatotoxic oligo, they just weed that out and they don't develop it further. In order to detect hepatotoxic oligos, if you're using gapmer ASOs, I think pretty typically people will give about 25 to 50 mg per kg of their oligo twice weekly for 2 weeks. And if you're seeing elevations in AST or ALT with that, then you'll typically not take those compounds forward into development. And that's usually done in mice. Now, mice are not as sensitive for detecting renal toxicity of nucleic acids. And so usually rats are used for that screen. Now, some of the issues that we see again and again in the published literature with immune evaluation of RNA or DNA, where people have said they didn't see any immune effects. One is failure to use an appropriate control. In the RNAi literature especially, there's a widely available commercial control RNAi that just happens to lack any of the immune stimulatory sequences that trigger TLR7 and TLR8. And I'm sure what happened is people just screened different controls and they found one control that gave results that they liked, and that's the one they use. You need to use a positive control in order to make sure that you can detect that. And it should be an appropriate TLR control, as I mentioned here. The mouse model is lacking functional TLR8, I mentioned already. Looking at the wrong time point. So I've often seen in the literature papers with RNAi formulated in LNPs in rodents where people just look at a 24-hour time point and they don't see interferon induction and they conclude it's not activating immunity. But in our hands, at least, by 24 hours, the serum cytokines are often back down to baseline. You really need to look at 3 or 4 hours in order to detect that induction. And then looking at the wrong readout. As I mentioned before, if you're using cell lines that don't express TLRs, you're not going to detect TLR activation. Okay, now another point worth mentioning is that there will be Antisense oligos and RNAi oligos are usually dosed at doses much, much larger than are required for TLR activation. In humans with CpG DNA, for example, a dose of 100 micrograms is enough to induce systemic CXCL10 induction in the serum. In antisense applications, you're typically giving hundreds of milligrams. And so there's a huge difference there. With the RNAi also, of course, RNAi doses that can activate TLR7 or TLR8 are much lower than the doses that are required to induce an RNAi effect, typically. Furthermore, these nucleic acids are internalized into endosomes, and so they can, they're in the right compartment to trigger the TLRs. And so the point I want to make is that immune effects are actually more likely to occur, and aptamer effects also, than antisense or RNAi effects are because of pharmacology of the oligos. And so it's just something to keep in mind when you're performing these experiments. Now, let me turn to human immunology and talk a little bit about what can be seen in humans typically as a result of these pathways. One of the most common effects seen in human antisense clinical trials very early on were the so-called flu-like symptoms and injection site reactions, or ISRs. And those are typically fatigue, fever, chills, things like that. Let me see. I see. Actually, this slide, I'm going to just make that point with them, but the next slide is the one I want to talk about next with more serious signs, the flu-like symptoms being relatively minor. Now, you hope that you're not going to see these. They're especially common with lipid nanoparticles or oligonucleotide conjugates. But what you can get are more severe manifestations than just the flu-like symptoms and injection site reactions. And that's hypotension, cytokine release syndrome. Back pain is also common sign of this. People don't often realize that. You can get flushing, swelling, fever, and these actually can occur immediately during injection. While the needle is still in the patient, you can have these symptoms come on. They can be that rapid. TLR activation pathways don't act that rapidly. It takes a couple of hours for things to come up. And so if you have that rapid an onset of an immune effect, there's really two major mechanisms that you need to consider. If it occurs on the first dose, you should consider complement activation. And oligonucleotides do activate complement. In primates, in the very first monkey studies of ASOs, phosphorothioate ASOs, complement activation and hemodynamic collapse occurred, and because people didn't realize that rapid IV administration of phosphorothioates can activate the complement cascade, and that can cause hemodynamic collapse. You can test for that using some of the markers I've listed here on the slide, C3A, C4D, and so forth. And you can also reduce the complement activation by slowing the infusion that you're giving. On subsequent doses, if it's not the first dose, what can happen is that you may be inducing IgE antibodies to the drug. It takes several doses, several exposures to the drug before these antibodies are produced. And an example of where this happened was a conjugate of PEG with an oligonucleotide. Regado was the company developing that. And they had a few cases of anaphylaxis, very severe inflammatory type 1 hypersensitivity syndrome occurring with that. And that appears to have been due to this IgE effect. Not very common, fortunately, in our field. And there are specific tests that you can do to check for that. If it's not immediate, but it's more delayed, there are a variety of possible mechanisms. Of course, within a few hours of injecting a nucleic acid that activates the TLR or the other innate immune pathways, you can get high levels of interferons and chemokines in the blood. Depending on the formulation and the exact nucleic acid, these can come up as early as 2 hours in humans. to around 24 hours in humans. So in that first time window is when you can see those coming up in the serum. Now, if it's just on the first dose, there's also what have been called idiosyncratic reactions. These can occur with monoclonal antibodies, with a lot of different biologics. They are not well understood, and usually they're just managed by slowing the infusion reaction. And then later in dosing, responses or reactions like this can be due to anti-drug IgG antibodies. And I did want to make the point that with ASOs, the phosphorothioates especially, anti-single-stranded DNA antibodies are relatively common. They're mostly not sequence specific, and they're usually not of pathologic significance. But when people test serum from patients, you may pick those up, and it's usually not clear whether they're relating to responses that the patients have or not. Anti-double-stranded DNA antibodies are what people tend to worry more about because these have been associated with lupus. Now, the normal screening assay for anti-double-stranded DNA antibodies, or dsDNA, is an ELISA assay, and it has a fairly high false positive rate. And so if you're doing clinical development of nucleic acids where you find a positive result by that assay, you ought to perform a confirmatory test using a more specific assay, and there's a couple of them here, so that you don't cause unnecessary concern with regulatory authorities that your drug might be inducing autoimmunity. Now, I wanted to make a few comments on formulations as well, and make a point that when you're using formulated nucleic acids, you have now two components of your drug that can be interacting with the immune system, the formulation and the payload. And we actually have some interesting data where the same formulation, Smarticles, has been used in humans by 3 different companies, Pronai, Myrna, and Mina. And in the case of Myrna, they were using this formulation to deliver an unmodified RNA. As they saw in the clinic, and this is all public information, was fairly significant cytokine release syndrome that they had to use pretty high dose steroids as prophylaxis for patients to give that safely. The same formulation with a modified RNA by Mina apparently has been well tolerated. And the same formulation with the DNA being developed by Pronai, they had some mild to moderate symptoms. And so the point here is, it's probably obvious, that the same formulation, you can reduce the immune effects if you're modifying your payload. And as I mentioned before, it's at least theoretically possible that different formulations may deliver the same nucleic acid into different intracellular compartments. And that's kind of the flip side of this. So that could have different immune effects. One more point I want to make, if you are working with PMOs, the morpholino oligos, you don't have to worry about most of this because the morpholinos have a neutral backbone, and that seems to avoid interaction with the innate immune pathways. They don't appear to activate complement, they don't activate TLRs. Now, because they have a neutral backbone and they don't bind proteins, they're also excreted very rapidly. But I did just want to make the point that what I'm talking about today with innate immune recognition of nucleic acids is really more for the phosphorothioates and the various flavors of phosphorothioate modifications. So I've listed here some references, excuse me, for further reading, and these slides will, by the way, be available. And just want to end with this comment that the immune system has had hundreds of millions of years to evolve these various ways to detect nucleic acids, and it's very good at it. We still have a very incomplete understanding of how these pathways work. We're still learning, we're still identifying new pathways and learning how they fit together. There's an awful lot that we just don't know yet. These species-specific differences especially are very incompletely understood. And what I've tried to do today is to give you an overview of all of this. I'm sure some of the things I have said are wrong or incomplete, and you'll just need to go from here and what we've talked about today to have a high index of suspicion for them, and hopefully we'll learn a lot more in the coming years. And finally, for anyone who wants to learn more about this space, I hope you'll consider coming to the Oligo meeting in Seattle, September 30 to October 3. It's an outstanding meeting covering all aspects of nucleic acid development, from preclinical to clinical, basic science, chemistry, formulations, delivery, everything you want to know. And now I'll be happy to take some questions. And I do see a question: if the immune response is different between human PBMCs in vitro and rodent models in vivo, what data should we trust in RNAi drug development? Well, that's a really good question. You know, I think we have to look at all of these assays, and ultimately we see what happens. And we've seen again and again examples of toxicities in humans that were not predicted in those. I think we have to screen for what we know to screen for, but ultimately in humans we still have to be careful, because there's any number of things that differ between humans and mice, and primates for that matter too. And there have been just too many different oligos that have gone into humans and caused unexpected effects and toxicities. So be humble in developing these. How can we screen for immunity? Wow. You know, that's not something I've had to do personally. In the brain, there are also innate immune cells, glial cells in the brain, supposedly express some TLRs, and they seem to be of monocyte origin. I don't know of specific screens for CNS responses, but I would start with the same screens that we're performing here, because they should detect immune activation of your compound. And I think you'd want to avoid it in the CNS. And I think doing CNS preclinical work in non-human primates as well as rodents would also help to reduce toxicities. Let me see, lots of great questions coming. Let me just come back up here. What doses do we screen in vitro? Oh yes, I didn't mention that. I would cover a range of doses in vitro, and it depends on whether you're using a delivery system or not. So there's so-called gymnotic delivery or free uptake, where you simply add oligonucleotides into the culture. This is with DNA oligos, and that's typically at concentrations of 10 micromolar or so. If you're transfecting oligos into cells, then you're probably going to be using nanomolar concentrations, but it depends so much on the specific chemistry and the formulation that you're using there, that I would look in the literature actually for references doing something similar to what you're doing and try to mimic that. I'd certainly want to go to concentrations that are higher than what you expect to use. But one point I should make is that concentrations in vivo, when you inject 100 milligrams or 200 milligrams of an ASO or an RNAi in vivo, the local concentration is going to be very, very high. It could be in the hundreds of micromolar concentration. And we are normally not screening those concentrations. So that's a deficiency in our in vitro screens. Design criteria for avoiding immune response, other than avoiding CpG and G-tetrads. The other main immune stimulatory motifs that people run into again and again are polypyrimidine sequences. So in general, several pyrimidines in a row, 3 or 4 pyrimidines in a row. I think TCT is one that people have identified. Those seem to be bad actors as well. There's some different information in the literature on that, but I do think polypyrimidines are another sequence class to be concerned about. Because non-primates can be costly early on, are there any small molecule models that can be used for testing in vivo TLR8 activity? I am not aware of that actually for TLR8 activity. We have just used human cells in vitro. I suppose you could use humanized mice, but they're not very cheap either. That might be one possibility. And then can I comment on mRNA drug? What are the difference from siRNA? So mRNA drugs are very likely to activate TLR7 and TLR8 by virtue of the stimulatory motifs in them. There are other cytoplasmic receptors that may be activated by mRNA drugs, but we really don't understand those very well. I think the TLRs are the ones that we understand the most. And delivering mRNA drugs and avoiding that, people are typically doing this by modification of the RNAs. There's a lot in the literature on that. Of course, different companies have different approaches to it. Sense optimization is an approach that CureVac has been taking, I believe, and they have reported nice results with that. Moving down the list here, compared to TLR9 in deoxy, what are the mechanisms successfully used for RNA-based mechanisms in the clinic? Mono asked that. Deoxy wasn't used success. Yes. Okay. So, the Mono is referring to there, TLR9 activation by CpG oligos has been therapeutically useful. For activating RNA in the clinic, mRNA vaccines actually, I think, are showing some encouraging results for cancer application, where you encode tumor antigens in an mRNA, and the TLR7 and TLR8 act like a built-in adjuvant to enhance the immune response to the protein. Now, one of the issues with this, though, is that when you inject your mRNA in vivo, mRNA is rapidly detected by the TLRs, and you get interferon production. Now, interferon production suppresses gene production. And so you have a little bit of a struggle between the innate immune system trying to shut down what it thinks is an invading organism, and your desire to get enough protein made so that you can induce an immune response to that protein. And so too much interferon production by an mRNA will actually suppress the production of the protein that you want to drive an immune response to. Nevertheless, mRNA vaccines clearly work. Moderna and several other companies, BioNTech recently has presented positive data with those as well. I think that's an exciting, very exciting area. And of course, the rapidity with which you can produce those vaccines is another really important aspect of them. Okay, how about PNAs? Could they activate immune systems since they're neutrally charged? No, that's correct. PNAs do not seem to activate immune response. Like PMOs, the PNAs really are, I think, a very attractive platform for development of nucleic acids that aren't going to have these issues. And I think in the long run, those kinds of neutral nucleic acids could be very therapeutically interesting if we can just get enough of them into cells to have more of an effect. If rodent studies don't predict human results, which way do the arrows go? More false negatives or more false positives? Actually, it can go both ways. We've seen that we had compounds that were toxic in rodents, but were not toxic in primates, and vice versa. So it can go either way, and it's hard to predict. We tend to do the rodent studies, and usually you kill anything that's toxic in rodents and you don't take it into primates, so we just don't know that. How much of the immune response is due to the PS backbone? Is there still a sequence component? There's definitely a big sequence component. The PS backbone, yeah, that's the PS backbone gives you very high protein binding. And with a PS backbone that is fully 2'-methyl modified, So that shouldn't be able to interact with the TLRs. And yet, we know that in the clinical trials done with exon fully 2' methyl modified oligos, there were effects occurring. There were chronic injection site reactions occurring, and acute injection site reactions as well. And yet in the in vitro assays that we've performed and that others have performed with fully modified methyl oligos, you don't see immune stimulation. But I think the problem there is the limitation of these in vitro assays, because typically we're only going up to 10 or 20 micromolar concentration, whereas vivo, in that local site, you're injecting hundreds of milligrams of your phosphorothioate, you have a very high local concentration, way above what we're normally testing. And so the biology of what's happening in those conditions hasn't been explored. But there's definitely a big sequence component to it. And I just point to the CpG oligos for that. So a good CpG oligo, 100 micrograms with a phosphorothioate backbone in the human, that's going to induce CXCL10. Whereas without the CpGs, if you avoid that, you have to still, without a CpG in an oligo, you can still activate TLR9 with a phosphorothioate. The phosphorothioate causes enough non-specific protein binding that you can activate these TLR pathways without a CpG, or if the CpG is methylated. Methylated CpGs on a phosphorothioate backbone do not completely eliminate the TLR9 activation, and I have a slide on that right here. So what you're looking at here is, this is stimulation index we're measuring in 293 cells, and with a positive CpG oligo control, we get decent activation of the NF-κB reporting. If you invert the CpG to a GPC, that knocks it way down, but not to zero. And if it's methylated CpG, it's intermediate. So methylation of CpGs in phosphorothioate oligos does not completely block TLR9 activation. It's commonly done because you also get a little bit higher affinity, but you can't assume that that completely does away with the TLR9 activation. Would you expect to see any immune stimulation in intrathecal administered oligos? The eye is an immune privileged site. There's not very many immune cells in the eye. And there have been, of course, intraocular oligos developed. I don't know what was seen in those. I wouldn't think it would be, I don't know. I just haven't done those studies myself. There have been reports, I believe, of TLR3 activation from oligos in the eye, but I'm not 100% sure about that, actually. Okay, let's see. Part of the reason that some chemicals such as PEG elicit an immune response is that they've recently become more prevalent in the environment and in medical treatments over the past few decades. How can we account for this and anticipate this issue in screening and drug development process? Wow, I don't know about PEG responses there. And I don't know how common an issue it is with PEG either in terms of causing these reactivities. The experience that Regato had was certainly very striking and a wake-up call for people that it's not innocuous putting PEG onto all of those. But I don't know the answer to that. I don't know how big an issue that's going to be. How much of immune stimulation is explained by the standard motifs? You know, people mostly avoid the standard motifs now. And so I don't think you're going to see nearly as many oligos going into clinical development that have CpGs. I think now what's more common that we see are oligos that have, for example, a TPG. So with a phosphorothioate backbone, a TPG also can cause TLR9 activation. And I think I have a slide on that here. Yeah, so, or this is even just poly-T. So I mentioned before that polypyrimidine sequences on a phosphorothioate backbone can have stimulatory effects. And that's especially true with poly-T, but you can mix some Cs in there as well. What you're looking at on this slide is on the top we're assaying for NF-kappa B induction, and this is on human PBMCs. And at the bottom, we're assaying for IRF7 activation by looking at interferon induction. Now, classically with CpG oligos, you activate both of these. And so the oligo on the right-hand side here, this is a CpG oligo, and it activates both of those pathways. If you have an oligo with a TCT right here, just and no CG at all, so we're just changing the G, the TCG to a TCT. You completely eliminate the interferon induction. You're not getting IRF7 being turned. You still get pretty good NF-kappa B induction, so you're still getting IL-10. You get rid of that and go to a T, and you get a lot less activation. You know, the TCTs, those are stimulatory effects that come up again and again in people's toxic sequences. And I think it's probably because of this non-canonical activation of TLR9, which is not that widely appreciated still. So I think these sequences are sequences that people have to be more aware of. Let's see. If you ignored immune activation when designing a fully modified PSASO, what's the probability that it would fail a clinical trial due to immune activation? That's going to depend on how much of it you have to give and how chronically you have to give it. And I'll come back here again to the experience of Prosensa with their exon skipping oligo for, that was a 2'-O-methyl, not a MOE, but I think it's probably going to be similar there. If you have to give 4 to 6 mg per kg weekly of a fully modified phosphorothioate oligo, you're going to get some of these effects, probably. I mean, maybe there's a way to select a sequence that's not going to do it. But what happened in these boys was that there were significant chronic injection site changes in the skin and some renal effects as well. And that was a sequence that, at least in vitro, just didn't see anything. It didn't seem to have any effects by the standard assays. They just didn't predict it. So again, you know, we don't fully understand the immunology of these compounds well enough to be able to fully predict this. And I would be a little worried about that potentially happening. Have you seen serious toxicities in patients receiving CMP-01? We did have one dose limiting toxicity in the study, and I think 13% of our patients have had hypotension following treatment. When you inject a CpG oligo, and especially a CpGA oligo, which is optimized for inducing high levels of interferon, inducing a lot of interferon in the subject is a stress. It's a physiologic stress. And the body normally responds to that by releasing more endogenous steroids to manage the stress. Where we've seen the most toxicity and the most hypotension are in patients who had adrenal suppression, where because the adrenal glands, they're not able to mount a normal stress response. And unless you give them stress dose steroids, they can get very sick very quickly. So we now know this in the clinic, and as soon as we have patients who show any signs of that, we give them fluids, we give them stress dose steroids, and it's actually been very manageable, all things considered. Certainly much less toxic than CAR T cells or some of the other therapeutics in oncology. But we're still learning more there too. Immunogenicity guidance is based on protein therapeutics. Regulators have asked us to do tests for our oligomer therapeutic. You bet they will. Could the cytokine, etc. tests proposed here be acceptable in lieu of antibody tests? Probably not, although I'm not a regulatory expert. I think that if you're developing an oligotherapeutic, the regulators are probably going to want you to design and develop tests for antibodies to that therapeutic. What you'll want to do when you're developing those tests is to, if it's a DNA, you'll definitely want to keep in mind what I said about the difference between anti-single-stranded DNA antibodies and anti-double-stranded DNA antibodies. Anti-single-stranded DNA antibodies in humans aren't that big a deal, and I don't think regulators get too concerned about them. but they'll probably want you to test for anti-double-stranded DNA antibodies too. And if you do that, you'll just want to make sure that if you use the ELISA assay, which is usually what people use because it's the simplest thing, you're probably going to get some false positive results with that. And so you'll want to use a confirmatory test. The Crithidia DNA assay is a good one. It's much more specific. And so what you'll find probably is that the positive results that you get with the anti-dsDNA ELISA assay are probably not going to be confirmed by the Crithidia assay test. And that is a reassuring thing for regulators. Can an external immune stimulatory event, infection, or allergy be exacerbated by the immune response to ASO? It would stand to logic that it could, I think, but I don't know of any literature on that, actually. I'd have to think more about that. There's nothing that comes to mind on it right now. There's so much heterogeneity and variability among humans. Not only are we incredibly genetically polymorphic, but as you're pointing out there, people come into a clinical trial with different basal states. Maybe the microbiome is involved too. It probably is. It seems to be involved in everything else. Can an in vitro assay for ISR? I'm not aware of any. I guess if your in vitro assays show cytokine induction, then you're probably going to get ISRs. But honestly, almost all oligo to have some ISR anyway, although it does vary a lot from one to another. I would imagine that oligos that induce more interferons or more cytokines in vitro are going to be more likely to induce ISRs in vivo. I guess we're running out of time, aren't we, Petra? Lots of great questions here, and I'm happy to up questions that we didn't have time to address here. I can do a few more, though. Let's see. Oh, good. Someone had a review of mRNA immunogenicity. Any data available on immunogenicity of tricyclo oligos? I'm not aware of that. Can you comment on how to distinguish immune system activation due to oligos versus due to the vehicle if the oligos necessarily needs the vehicle to delivery? I don't know how you can easily do that. I think you really have to look at the two of them together in that case. And then mono, I was thinking about short TLR7, 4, and 8, and RIG-I. What are the challenges in making use of them as adjuvants compared to CpG oligos? We just haven't done much with the short RNAs as adjuvants, actually, compared to CpG oligos. CpG oligos work very well. I would think the others would work well, too. And I'm not aware of unique challenges with them. Is there a simple relationship, monotonic linear, between percent of oligochemically unmodified and immune stimulation? I would say, I don't know of any simple relationship, actually. I think one of the things that we have seen is that more than the percent of the oligo that's modified or unmodified is exactly where the modifications are. And just changing a modification from one position to an adjacent position can make a huge change in the immune effect of the oligo. And we really don't understand why that is. You just have to have a high index of suspicion for them. Why are some of the injection site immune responses not observed when going IV using full 2? I think the reason you don't see these responses IV is because you're diluting the oligo in the whole body. For example, with CpG oligos, I mentioned very small doses given subcutaneously. Actually, I didn't mention that. But with the CpG oligos, if you inject just 100 grams subcutaneously, you can detect serum elevations in CXCL10. You inject intravenously, and you don't see those elevations, even with much higher doses. So I think it's just a dilution effect in the rest of the body there. And maybe that means it's going to be safer for a lot of indications to give the oligos through the IV. But I guess we need to wrap this up, Petra.
Yes, so unfortunately Art has a hard stop in about 2 minutes. If you want to ask any more questions, please type them in the chat box. I will send them off to Art, and he's agreed to answer them writing or taping himself answering the questions at a later date. Hopefully you've all enjoyed the webinar, and thank you very much, Art, for doing it. That was very informative. Also, the next webinar we will be having is on basic principles of IP and intellectual property and patenting. Details to be found on the website soon, and we hope to see you there. Okay.
Oh, and the slides will be available.
And thank you, Art, and goodbye.
Thank you all. Thanks for dialing in today. Bye-bye.
Bye.